Hello students, I'll today introduce you to a topic in the microbial nutrition module, which is also a part of the module on bacterial metabolism, that is assimilation of nitrogen. Now, assimilation of nitrogen refers to the incorporation of inorganic nitrogen into organic material in the cell. If you remember, we have been discussing assimilation of carbon in our earlier lectures, the module bacterial metabolism. Here I'm going to present to you a brief of how nitrogen is assimilated by prokaryotes. Now, just like carbon, nitrogen is a macronutrient and you already know of its structural and functional role. It is a part of various biomolecules. The atmosphere contains a huge pool of nitrogen, but this is largely unavailable to living organisms. So it needs to be fixed for uptake by organisms, whether it is microorganisms or it is higher organisms. Now the triple bond in N2 actually makes it quite unreactive. The bond energy is of the order of 225 kilocalorie per mole. In nature, the fixation of nitrogen takes place by abiotic means, especially when, for example, lightning serves as a source of energy and nitrogen oxides are formed. This is not within the purview of the course. We will focus only on microbe-mediated processes. I'll take you through two sets of microbial reactions. The first one wherein atmospheric nitrogen is converted to ammonia, making it bioavailable, and then how this ammonia becomes a part of organic matter or specifically amino acids. Now, most nitrogen is fixed biologically by the process of nitrogen fixation. This process, which involves the reductive breakage of the triple bond of N2, leading to the formation of ammonia, is carried out exclusively by prokaryotes. And organisms which can perform such a process are called as diazotrophs. Biological nitrogen fixation, or BNF, is believed to contribute half, if not more, of all the global fixed nitrogen. Now, the biological process of nitrogen fixation requires a complex enzyme called as the nitrogenase complex, which carries out this transformation. Nitrogenase consists of two proteins, a reductase, which is an iron protein. This provides electrons with high reducing power, and the other component, or the nitrogenase component, which is a molybdenum iron protein, uses these electrons from the reductase component to reduce N2 to NH3. The transfer of electrons from the reductase to the nitrogenase component is coupled to the hydrolysis of ATP by the reductase. Now, the nitrogenase complex is extremely sensitive to oxygen and gets deactivated under aerobic conditions. In fact, this is the reason why in the nodules of legumes where rhizobium fixes nitrogen, a very low concentration of free oxygen is maintained. Of oxygen in the nodules. You know about the pigment leg hemoglobinules where rhizobium fixes nitrogen, a very low concentration of free oxygen is maintained. I'm not sure if you know about the pigment leg hemoglobin, which is a homologue of hemoglobin. This binds to O2 and brings down the concentration of oxygen in the nodules. Now, there are three main taxa of nitrogen fixers: bacteria, archaea, and cyanobacteria. And based on their ecology, there are free-living nitrogen fixers belonging to the genera, for example, Clostridium, Azotobacter, Bacillus, or Azospirillum. And there are symbiotic bacteria. The most popular of one which you know is of Rhizobium associated with legumes. But legumes are not the only ones in association with diazotrophs. There are associations even with non-legumes. For example, association of Frankia with actinorhizal plants and cyanobacteria with cycads or ferns, or even associations found in lichens. In the case of archaea, the process is found only in methanogens, if you see here. Again, we have talked about methanogens in one of our earlier lecture. Non-symbiotic bacterial diazotroph, in fact, contribute approximately 30% of the total biological nitrogen fixation. Now, this is how the net reaction of biological nitrogen fixation looks like, wherein N2 is converted to ammonia. The reduction of N2 actually takes place in 3 steps, each requiring a pair of electrons. So basically, the reduction of N2 to NH3 is a 6-electron process, 6-electron requiring process. However, the process always produces at least 1 mole of H2 in addition to 2 moles of NH3 for each mole of N2. Hence, an additional input of 2 electrons is required. Now, because the process yields molecular hydrogen as a side product, you can understand this is also a process of interest to scientists who are looking for means to produce H2 as an alternative energy source. Now, the iron protein component of the complex is first reduced by electrons donated by ferredoxin. Then the reduced iron protein binds ATP and reduces the second component, which is the molybdenum iron protein, which eventually donates electrons to N2. Now, in most diazotrophs, the 8 potential electron actually come from the reduced ferredoxin. And depending on which bacteria or microorganism we're talking of, the reduced ferredoxin which supplies electron for this process is generated by either photosynthesis or respiration or even fermentation. And in this process, 2 molecules of ATP are hydrolyzed for each electron transferred. So at least 16 molecules of ATP are hydrolyzed for each molecule of N2 reduced. So you see this is an expensive process. In addition, the enzyme complex nitrogenase can reduce several other alternative substrates which resemble N2 on the basis of double or triple bonds in the structure, like acetylene, cyanide, or even azide. This is just to connect you with a process that we have covered in one of the earlier lecture pertaining to nitrogen metabolism. So while diazotrophs, they fix nitrogen to form ammonia, it is nitrifiers, the process of nitrification, if you remember, which converts ammonia to nitrate. And I already mentioned to you that both ammonia and nitrate are forms that plants or higher organisms can utilize. Now, just to remind you, we've also come across a process which leads to conversion of nitrogen oxides to atmospheric nitrogen, thus leading to a loss of nitrogen from agricultural system. This process is termed as denitrification. Now talking of the next set of reactions wherein N is assimilated in biomolecules, and we will focus on its incorporation into amino acids. The uptake of N-containing compounds in a prokaryotic cell could be either through diffusion or it could be through active transport, and these compounds are subsequently converted to ammonia. Ammonium assimilation is mediated by two pathways, either by glutamine synthetase, this takes place at low ammonium concentrations, or by the glutamate dehydrogenase system, which functions at high ammonium concentrations. And the glutamate and the glutamine thus generated, they serve as major amino donors for all assimilations or incorporation into other cellular components. Now we'll take up both these pathways, starting with GDH pathway or glutamate dehydrogenase. This is a pathway, as I mentioned, it is active under high nitrogen supply. And here I'm referring to ammonium concentrations of 1 millimolar or above. This is because the affinity of GDH to ammonium is too low. By this pathway, one net glutamate is generated by the reductive amination of 2-oxoglutarate. So if you see here, 2-oxoglutarate is being converted to glutamate, and the process, it does not involve any ATP, but 1 mole NADPH is oxidized per mole ammonium assimilated. This is just to show you the chemical structures and the amination involved. If you see, this is alpha ketoglutarate taking up an ammonia and forming glutamate, which is an amino acid. Now, at ammonium concentrations below 1 millimolar, the affinity of GDH is towards ammonia is too low, and so the other pathway takes over. This is mediated by this high affinity cyclic GS Gogart pathway, glutamine synthetase, glutamate synthase pathway. GS stands for glutamine synthetase and Gogart for glutamate synthase. This also produces, similar to the earlier pathway, one net glutamate by reductive amination of 2-oxoglutarate. This also produces pathway. GS stands for glutamine synthetase and Gogart for glutamate synthase. This also produces, similar to the earlier pathway, one net glutamate by reductive amination of 2-oxoglutarate. However, here one ATP is invested for every amino group assimilated. As the name suggests, it's a synthetase. Now, GS or the synthetase has a relatively high affinity for ammonium, while GDH has a relatively high Km value for ammonium, which is approximately 1 millimolar. GS has a 10-fold lower Km for ammonium, that is 0.1 millimolar. Now, to economize the use of ATP, as you can understand, a cell downregulates the GS Gogart pathway when ammonium is present at high concentrations. Now, both GDH and Gogart, they are specific for NADPH over NADH. Again, this is to show you the chemical structures of the components involved in the pathway. So if you see, this is the glutamine synthetase, which takes up ammonia, converts glutamate to glutamine, and glutamine serves as another amine donor, a strong amine donor. Basically, here you see glutamine transferring its amino group to another 2-oxoglutarate, ultimately resulting into one glutamate, net one glutamate, because the other one goes into the cycle. It's a cyclic pathway. Now, one ammonium is incorporated as once this happens, that it incorporates as glutamate or glutamine, as I mentioned, both these moieties can serve as donors of amino group to form further amino acids and other biomolecules by the action of transaminase. In fact, Gogart that you see here is also a transaminase. In fact, the name is glutamine oxoglutarate from the substrates, and it's called glutamine oxoglutarate aminotransferase. Before I end, just to inform you that with today's lecture, we have completed the entire module of bacterial metabolism and nutrition. In the process, we have in fact discussed the three major steps of nitrogen cycle, that is denitrification, that is generation of N2. Then we've talked about nitrification, wherein ammonia is converted to nitrite and nitrate. And today we talked about nitrogen fixation. So you can see that this entire cycle is majorly microbe driven. And here you can clearly see how there's a demarcation between the oxic and the anoxic processes.